Ever think about where all those like flavor enhancers come from? The stuff that's in like almost everything you eat, or the ingredients in animal feed. It's all happening on a crazy big scale, industrial scale. But what's really cool is that scientists are like figuring out these microbial tricks, these ways to really amp up production of some of these like everyday things. And that's what we're going to get into today.
Yeah, that's right. We're going to explore how scientists have learned to improve, well, actually overproduce is the better term, some really valuable primary metabolites from microorganisms. Our sources this time, they really get into some fascinating ways that these tiny organisms are being tweaked to just be super efficient production machines, tiny little factories. It's like getting a peek behind the curtain of industrial biotech.
So basically, our mission today is to understand the core strategies behind strain improvement,
Yeah.
right? Like how they actually do it. We're going to try to keep it clear, like cut through the jargon, so you can really see the insightful techniques that they're using. So, okay, how about we start with something I bet everyone listening to this has encountered. L-glutamate, the thing that makes MSG. MSG.
Absolutely. And the key player here is a bacterium called Bacterium glutamicum. And one of the early and really clever things they figured out was to use auxotrophic mutants, specifically homoserine auxotrophs. So you might be thinking, why would blocking homoserine synthesis lead to more glutamate? It's actually pretty cool. It forces the metabolic flow, the inner workings of the bacterium, to shift. By creating this sort of bottleneck, it makes the organism channel more resources into making glutamate. A good example is C-glutamicum ATCC 14067.
Hmm. That makes sense. Like redirecting traffic. If there's a jam, you close one road to open up another. What about these feedback-resistant mutants? You mentioned those before. What's going on there?
Well, imagine a cell. It doesn't want to just keep making more and more of something if it doesn't need it. It has these built-in kind of stop signs, feedback mechanisms that regulate how much it produces, feedback-resistant mutants like C-glutamicum KY9102, for example. They've been modified, so they basically ignore these stop signs. They just keep overproducing, which is like
Wow.
a gold mine if you're trying to produce this stuff on a massive scale. I guess you could say it's kind of like taking the brakes off a production line. You just let it run and run.
Wow, that's wild. And you also mentioned permeability mutants. What are those all about?
Oh, these are fascinating. They've basically been engineered to let go of that glutamate more easily, to release it into their surroundings. What this does is it makes the whole extraction and purification process, like downstream, way more efficient. It's like they're not only producing more, but they're making it easier for us to collect it, which is amazing when you think about it.
Okay, let's switch gears a bit. Let's talk about L-lysine, another important product from our friend Bacterium glutamicum. And this stuff is crucial for animal feed and fortifying our food too. What tricks did they pull out of their sleeves to boost L-lysine production?
Well, with L-lysine, one strategy uses mutants that are resistant to this compound called diamine ethylcysteine, DAEC. This resistance often messes with an enzyme called aspartaginase, which is normally kept in check, inhibited by lysine. So by becoming resistant to this inhibitor, the bacteria basically bypass a major control point in lysine productions, like rewiring the system. And C-glutamicum AJ3456 is a really good example of that.
And there was an even more efficient method. You were talking about threonine exotrophy being used with DAEC resistance.
That's right. There's a really interesting synergistic effect that happens there. You can see this in strains like C-glutamicum RB41. So they're not only resistant to DAEC, they're also unable to make threonine, another amino acid. So basically, their metabolic machinery is even more focused on pumping out lysine, a really clever way to maximize output.
That's wild.
Yeah.
Like these tiny little cellular tweaks can have huge real-world effects. Pretty amazing. All right, let's shift focus one more time. Citric acid, that's produced in massive quantities by Aspergillus niger. It's a fungus. What are the key tricks they're using there?
So with Aspergillus niger, well, researchers discovered that sometimes resistance to certain substances correlates with an increase in citric acid production. Like methanol, some methanol-resistant mutants, they often produce way more citric acid. Same thing with fluoracetate. Mutants that are resistant to it, they often have reduced activity of an enzyme called aconitase, which leads to citric acid buildup. It's almost like if you partially block a step in the metabolic pathway, further down the line, you force the flow to go where you want it to make more citric acid.
No, that makes sense. Like a dam,
Yeah.
a controlled overflow.
Yeah.
And you also mentioned using radiation to make mutations. That sounds like a way less targeted approach.
Yeah, it is. They've used UV and gamma radiation to basically randomly cause mutations in Aspergillus niger.
Yeah.
And then what they do is they screen the offspring, to find the ones that just happen to produce more citric acid. It's definitely a throw a bunch of stuff at the wall and see what sticks approach compared to targeted modifications. But, it's worked. Historically, it's led to some really beneficial mutations.
Okay, so looking back, I mean, we talked about glutamate, lysine, citric acid. Are there any big picture, like overarching strategies, things that you see popping up over and over?
Definitely. A few themes keep coming up. Like one is targeting those natural regulatory mechanisms in the cell, like feedback inhibition and repression. Another is the whole idea of engineering metabolic pathways through mutagenesis, whether it's super precise or more random. It's a common thread. And then there's using oxytrophy to redirect those metabolic resources. We talked about that. And lastly, selecting for resistance to certain analogs or inhibitors. That's a big one. And the thing is, it's often iterative. You modify, select, modify, select. It's a cycle that leads to more and more efficient strains. It's pretty amazing.
So listeners, what we really want to drive home is just how powerful these seemingly tiny tweaks to microorganisms are. They have huge industrial implications, and it's just mind-blowing how fiddling around at a cellular level can lead to such big increases in production. I mean, this is stuff we use every single day.
It really is incredible. What's really fascinating to me is the ingenuity behind it all. It's not just, randomly experimenting. It's a really deep understanding of microbial metabolism. They're finding really clever ways to optimize it to meet our needs, which is super interesting. It makes you think.
Yeah, I'm definitely looking at the products I use every day in a new light now. So thinking about all these successes, like with mutation and selection, what new possibilities are there? Especially with all these advanced genetic engineering techniques coming up, what does the future hold? It's exciting to think about how we might be able to refine these tiny biological factories.
Oh, absolutely. It's an area that's just bursting with potential.
Oh.
Who knows what we'll discover
It's
next?
amazing to think about. All right, everyone, thanks for joining us for another deep dive. We'll see you next time.